We have already discussed how antigen is processed in cells and how it is presented on cell surface with HLA molecules. Now we will see how HLA molecules interact with cells of adaptive immune response on cell surface and how immune response is organized according to nature of pathogen. Immune response for a pathogen depends on nature of that pathogen. So immune response for a virus is completely different than immune response for extracellular bacteria. So how immune system recognize which pathogen it is dealing with? This is achieved by recognition of molecular patterns of pathogens by antigen presenting cells. These molecules are present on surface of pathogens or can be their other components including their DNA and RNA. These molecules are called pathogen-associated molecular patterns. These molecules are different than self and even not present in human beings, like peptidoglycan of bacterial cell wall. So whenever antigen-presenting cells will come across these molecules, they will immediately recognize that they are dealing with something that is not self, and an immune response will potentially be initiated. These molecules are usually necessary for survival of pathogens, like components of their cell wall. So there will be no significant change in these molecules over time. Another important characteristic of these molecules is that they can be common to a group of pathogens, like lipopolysaccharides are component of cell wall of gram-negative bacteria. So whenever antigen-presenting cells will come across lipopolysaccharides, they will recognize that they are dealing with a gram-negative bacteria. Similarly, other molecules will help in identification of other groups of pathogens. So it is the molecular patterns of pathogens that are identified by antigen-presenting cells. Next question is how antigen-presenting cells recognize these pathogen-associated molecules. These molecular patterns are recognized by antigen-presenting cells by receptors on their cell surface called pattern recognition receptors, like toll-like receptors. These receptors are also present in cytoplasm and endosomes of antigen-presenting cells. So whenever a pathogen or molecules derived from pathogens will come across these receptors, antigen-presenting cells will recognize their molecular patterns to identify nature of pathogen. So when antigen-presenting cells like dendritic cells ingest a pathogen, peptides derived from degradation of this pathogen are presented on HLA molecules on surface of antigen-presenting cell. Other molecules derived from this pathogen are recognized by pattern recognition receptors in cytoplasm and endosomes of these cells. So at the end of this process, dendritic cell will have presented peptides derived from this pathogen on HLA molecule on cell surface and will also have recognized and identified which pathogen it is, whether it is a gram-positive bacteria or a double-stranded DNA virus. Now, what happens after pattern recognition receptors of antigen-presenting cells have recognized molecular patterns of a pathogen? This results in downstream signals from these receptors that will result in production of cytokines. This will also result in expression of surface molecules on dendritic cells, including expression of B7. This expression of B7 means that dendritic cells have identified an infectious agent. Dendritic cells then move from site of infection to regional lymph nodes, where they will present antigens to CD4 T cells. In lymph nodes, dendritic cells present antigens on HLA class II molecules to naive CD4 T cells. CD4 T cells are the cells that organize further immune response. However, this is not a single-step process. A series of signals is required for activation and proliferation of naive CD4 T cells. First signal for activation of CD4 T cells is interaction of T cell receptors of these cells with antigen present on HLA class II molecules. If T cell receptors have sufficient affinity for this antigen, then CD4 co-receptor of CD4 T cells bind to HLA class II molecules. This interaction of CD4 co-receptor with HLA molecules has three functions. It helps CD4 T cells to recognize HLA class II molecules. It also provides some stability to this interaction. And finally, some downstream signals are sent from T cell receptor complex to T cells. These signals result in initiation of activation of T cells. However, this activation will not go any further unless there are additional signals. If we compare this with interaction of effector CD8 T cells with cell presenting abnormal antigen, in this case, these two signals are enough for activation of CD8 T cells. So interaction of T cell receptor with antigen on HLA class I molecule and then interaction of CD8 co-receptor with HLA molecule is enough for activation of CD8 T cells. CD8 T cells then release cytotoxins like perforins and granzymes that result in lysis of cell presenting abnormal antigen. However, for further activation of naive CD4 T cells, further signals are also required. Second signal required for activation of CD4 T cells is interaction of B7 molecule present on dendritic cells with CD28 molecule present on CD4 T cells. We know that B7 is expressed on dendritic cells only when they come across foreign antigens, while CD28 is even present on naive CD4 T cells. This signal is called co-stimulatory signal and it is required for further activation of T cells. Importance of this signal can be recognized when this signal is absent. If there is peptide present on HLA class II molecule but B7 is absent, then co-stimulatory signal will also be absent. This will result in energy of immune response, and although HLA molecules have antigen, there will be no further immune response to this antigen. Absence of B7 can also lead to differentiation of regulatory T cells and tolerance to antigen present on HLA molecules. Although there are other co-stimulatory signals present as well, as CD28 is present on naive CD4 T cells, so this is one of the first and important co-stimulatory signal. Now suppose if there is B7 and there is interaction of B7 with CD28, this will result in downstream signals from CD28 that will result in activation of transcription factors in CD4 T cells and expression of IL-2 gene and upregulation of IL-2 receptors on cell surface of these cells. This will also result in expression of other cytokine receptors on these cells and expression of CD40 ligand molecules on cell surface. CD40 ligand has role in antibody-mediated response, and its interaction with CD40 molecule present on B cells result in class switching of immunoglobulin molecules and other functions related to antibody-mediated response. So second signal result in production of cytokines, including IL-2, and upregulation of IL-2 receptors on surface of CD4 T cells. IL-2 acts in autocrine fashion and it is a potent signal for proliferation of CD4 T cells. IL-2 is not only a signal for proliferation of CD4 T cells, but it is also a signal for proliferation of CD8 T cells and B cells. For this reason, many immunosuppressant therapies are based on IL-2. Some of these medications block IL-2 receptors, while other medications block downstream signals from these receptors. Now we have seen process of CD4 T cell activation and proliferation. Now we will see how immune response is directed according to a particular pathogen. After activation and proliferation of CD4 T cells, a second wave of cytokines, also called third signal, will direct immune response according to nature of pathogen. This process actually starts when dendritic cells recognize molecular patterns of pathogens. Dendritic cells then produce cytokines based on their findings. Then interaction of dendritic cells and CD4 T cells in lymph nodes finally result in activation and proliferation of CD4 T cells. These CD4 T cells or helper T cells then will convert to subsets of helper T cells by these cytokines according to nature of pathogen. These subsets of helper T cells then mediate immune response depending on nature of pathogen. Suppose pathogen was an intracellular pathogen like a virus or Mycobacterium tuberculosis. Main response in this situation will be cytotoxic response, also called cell-mediated response. In this case, helper T cells will convert to type 1 helper T cells or Th1 cells. This is mediated by a cytokine interferon gamma that help conversion of helper T cells to Th1 cells. Th1 cells then in turn secrete other set of cytokines that help in activation and proliferation of effector cells of cytotoxic response. These effector cells result in lysis of infected cells in case of viral infection or formation of granuloma in case of Mycobacterium tuberculosis. Effector cells of cell-mediated response include CD8 T cells, macrophages, and natural killer cells. Cytokines that mediate this response include interferon gamma and IL-2. In lymph node, dendritic cell initially presented antigen on HLA class II molecules to CD4 T cells. However, dendritic cells can also present antigens on HLA class I molecules to CD8 T cells. This is called cross-presentation of antigen. This leads to activation and proliferation of antigen-specific CD8 T cells. So both naive CD4 and CD8 T cells are activated by interaction with dendritic cells. CD4 T cells organize immune response, while CD8 T cells are effector cytotoxic T cells. Cytokines that mediate Th1 type response also have inhibitory effects on Th2 type response. That is mainly antibody mediated response. So cytokine interferon gamma that mediates Th1 type response also has inhibitory effect on Th2 response. This does not mean that antibodies have no role in viral infections. Antibodies can have important role in controlling viral infection, like antibodies help in neutralizing viruses in extracellular compartment and also contribute to antibody-dependent cell-mediated cytotoxicity. This process of ADCC does not require CD8 T cells. Now suppose if infectious agent is extracellular pathogen, then main response will be Th2 response or humoral response, also called antibody-mediated response. In this situation, interleukin IL-4 will convert T helper cells to Th2 cells. These cells will then secrete other cytokines like IL-4, IL-5 and IL-13. These cytokines help in proliferation of B cells, class switching of antibody type, and conversion of B cells to plasma cells. Some of B cells will then convert to memory B cells, like memory CD4 T cells in cytotoxic response. Similar to Th1 response, cytokines that mediate Th2 response, mainly IL-4, have inhibitory effect on Th1 response. However, Th1 and Th2 response is not static, and nature of response can change during course of disease. In addition, other antigen-presenting cells, like B cells, can also present antigen from same pathogen to CD4 T cells that also result in antibody-mediated response. Other cytokines help conversion of T helper cells to other subsets of helper T cells. These subsets have different functions, including regulation of immune response. We have seen how immune response is initiated and how it results in activation and proliferation of T and B lymphocytes and production of cytokines and antibodies. All this is needed for clearance of infection. However, this process needs to be slowed down at some time, as activated immune cells and cytokines can be potentially harmful to host. This is immune response in graph form. Immune response initiates, and then it builds over days, then gradually declines, and finally only a memory response is left. So how immune response slows down after some time? This is achieved by different ways. One way is as soon as B7 starts activating CD4 T cells through CD28 receptors, CD4 T cells start expressing another molecule on cell surface called CTLA4. CTLA4 competes with CD28 for binding to B7 molecule. When CTLA4 binds to B7, this results in inhibition of activating signals being sent to CD4 T cells. That result in decrease in production of cytokines, including IL-2, and decrease in proliferation of these cells. There is another similar inhibitory pathway that involves PD-1 molecule present on activated T cells. This molecule interacts with PDL1 molecule on surface of dendritic cells. This interaction also results in inhibition of downstream signals from T cell receptor complex that result in decrease in production of cytokines and reduction in T cell proliferation. In this way, immune response slows down and then gradually wears off. In summary, immune response starts when antigen-presenting cells like dendritic cells come across pathogens. Dendritic cells detect molecular patterns of pathogens and also present peptide antigens derived from these pathogens on HLA class II molecules to CD4 T cells. These cells can also present antigens on HLA class I molecules to CD8 T cells. So dendritic cells present antigens to both CD4 and CD8 T cells that result in activation of these cells. From site of infection, dendritic cells then move to regional lymph nodes, where they present antigens initially to CD4 T cells. Resulting interaction of HLA molecules with T cell receptor complex and co-stimulatory signals result in activation and proliferation of CD4 T cells. Based on cytokines derived from antigen-presenting cells and CD4 T cells, immune response is then organized according to nature of pathogen. Finally, inhibitory molecules appearing on surface of T cells result in slowing down and finally turning off immune response.